Go. Here we go. All right. Kind of the last little concept I want to introduce is one that will give you some really great cred if you're ever at fancy cocktail parties. One of the best known papers in evolution was written, this is back in the 70s, by Stephen Jay Gould and Richard Lewontin. Uh, Gould was well known as a paleontologist. Lewontin is most famous for developing gel electrophoresis and using that as a means of gathering allele frequency data. But they were both men of a very philosophical turn of mind, and they wrote a paper, probably most famous for its title, The Spandrels of San Marcos, a critique of the Panglossian paradigm. And you will impress the heck out of people at cocktail parties if you say, oh, yes, the other day, yes, I was reading about how the spandrels of San Marcos are a critique of the Panglossian paradigm. What the hell is that? I'll break it down. Well, first of all, okay, this French author who wrote under the name Voltaire, his real name was Jean-Marie Arouet, but he wrote under the name Voltaire. And he wrote a book called Candide, which is very funny and very bitter and takes a lot of potshots at the philosophy of the time. And Candide is this innocent guy who has all kinds of adventures, most of them ending horribly, in quest of his beloved, the Princess Cunégonde. And Candide's teacher is Dr. Pangloss, which is Greek for all tongue. And Pangloss is firmly convinced that this is the best of all possible worlds. That this world, what looks like evil and pain really isn't, because this world is the best world of all of the possible ones that anyone could imagine. Anything other than this would be less good. So everything has to be happening for the best, turning out the best way that it is logically possible for it to turn out. And Pangloss keeps saying this after getting one of his buttocks chopped off and starving and getting leprosy, I think. And I think like getting syphilis and having his nose fall off or something like that. All of these absolutely rotten things happen to Dr. Pangloss. And he keeps saying that, well, this has to be for the best because we are clearly in the best of all possible worlds. So Panglossian optimism is optimism to the point of complete stupidity. Where he comes in here is that Pangloss keeps observing how this must be the best of all possible worlds because he keeps noticing how beautifully designed everything in the world is. So he notices, for example, that cows are clearly designed so that we can have beef all year round. Legs are perfectly shaped to fit into trousers. Noses are beautifully shaped so that they can carry glasses. Think about it. You wouldn't be able to wear glasses if you didn't have a nose of exactly the right shape in exactly the position where it is now. So clearly, noses are beautifully designed, part of this best of all possible worlds. And what Gould and Lewontin mean by the Panglossian paradigm is the idea that everything in living things is the product of adaptation, that everything is shaped by natural selection for the purpose that it has now and always has been. Their critique of it is based on something called a spandrel. What the hell is a spandrel? Okay. When you churches back 1,000 years ago, especially in the Byzantine or the Eastern Roman Empire and places influenced by that, very often use the device of putting a circular dome on top of four big arches. And if you do that, what you end up with is four curved triangular spaces that are usually filled up with wall in between the four arches. If you want to really get technical, an architect would call these pendentives and not spandrels, but it's mostly the same thing. A pendentive is a 3D variation on a spandrel. A spandrel is what's left over when you put an arch in a wall. So what do these look like? Well, that's from an architecture glossary that I found. There you've got an arch. It's supported on four domes. And right there, you can see that between two arches and the floor of the dome, you have this kind of curved triangular wall space. That's a spandrel, or that's a pendentive, but a pendentive is a type of spandrel. In side view, that's another one of the spandrels. From behind, that's another spandrel. Right there's a little corner of the fourth spandrel right there. I'm gonna say spandrel without worrying too much because I'm not an architectural historian. You know, we've got other departments to cover that. I don't care. Gould and Lewontin called them a spandrel. Okay, what's so great about spandrels? Well, here's the interior of St. Mark's Cathedral, San Marcos in the title that we mentioned, the spandrels of San Marcos. And they're famous because they're covered with golden mosaics all over. The windows aren't that big. You can see a few little ones up there. But every interior surface is covered with these golden mosaics. So here, you're looking up at a dome. Right up there in the center of the dome, that's Christ. You have various saints and angels surrounding Christ, looking up at him right there, lower in the dome. And right there is one of the four spandrels. And in the spandrel, that is something Christi Sanctus Matthäus. That's a picture of Saint Matthew, one of the evangelists, one of the authors to whom are attributed the Gospels, right? The author of the Gospel of Matthew in the Bible. And then over here, Discribuit quaterlisti. I can't read that, unfortunately. That's a little too blurry. But the guy sitting here, that's another one of the evangelists. And so you have these four evangelists who are down below the dome, looking up at Jesus, writing down what he's telling them to write. And you can't see them in this picture, but the other two evangelists, right? Matthew, Mark, Luke, and John are depicted on these four spandrels. So it's a really neat picture, right? You've got Jesus, you've got all the saints up in heaven, and then these four spandrels are perfect places to display the four evangelists looking up at Jesus and saying, wow, tell me more. Let me write all this down. And they're sitting at their desks writing the Gospels. That's another spandrel. This one, the decoration's been damaged, but this is under the dome of the Church of Hagia Sophia in Istanbul. There's the dome, and then this triangular shape here is one of the spandrels. I think it originally had an angel on it. Hagia Sophia, up until recently, was a museum. It was in the news when the premier of Turkey ordered it to be converted back into a mosque. It was built as a Christian church, but converted into a mosque when the Turks took Constantinople in 1453. Now it's Istanbul, not Constantinople. Been a long time gone. Constantinople, why did Constantinople get the works? That's nobody's business but the Turks. Good song. Okay. So here's my point. Spandrels are beautifully decorated in these old churches. They were often just covered in mosaics. Gould and Lewontin point out that, they're very convenient for impressing believers with the beauties of Christian faith. I showed you that picture inside St. Mark's Cathedral, and you have Jesus at the very top of the dome, surrounded by saints and angels, all surrounding him, all worshiping him. And then below the dome, you have these four spandrels that display mosaics of the four evangelists, Matthew, Mark, Luke, and John, looking up at Jesus and writing down their gospels by his inspiration. It makes a really nice picture. It's very visually impressive. It conveys the Christian faith. And yet, as handy as spandrels are, they weren't selected by the architect for the purpose of decoration. The architect didn't say, okay, I'm going to pick a design, I'm going to build my church for the purpose of displaying these beautiful mosaics. And I'm going to put the spandrels there so that they can display the four evangelists, or in another case, the four rivers that flow from the Garden of Eden. Those are also depicted on another set of spandrels in the cathedral. The point is that spandrels weren't selected at all. Spandrels exist because they have to. If you're very good at building domes and arches, like the Romans were, and they passed that knowledge on to later generations. If you're going to put a dome on top of arches, the triangular spaces just automatically appear. The architect didn't put the spandrels there so that they could hold those mosaics. The spandrels are there because they're just there, just out of the geometry of supporting a dome on arches. The architect didn't design the cathedral for the purpose of the mosaics. The architect designed the cathedral that way because it was a design that he knew would stay up. And then after the fact, the spandrels become useful for displaying the evangelists, but they're not the reason that the structure is there. Just like your nose is useful for holding up your glasses, but that's not why your nose is there. Your legs are useful for putting trousers on, but that's not the reason why you have legs. Your ancestors had legs for a very long time before any of them thought of putting on trousers. Gould and Lewontin, I'm sorry for this long digression into the history of architecture, which may not be the thing you're most interested in. Gould and Lewontin argue that many features of organisms are not adaptations, they're not selected for, they result from the same kind of geometrical necessity. I'll give you an example here. The shell of a snail is basically made by taking a cone and coiling it. Imagine taking a very long cone and making a coil out of it. You might make a very tight coil or a very loose coil. You might make a coil almost in one dimension, so you get a very flat shell, or a coil that moves through a third dimension, so you get a very pointy shell. There's lots of different shapes that you can make just by taking a cone and coiling it, depending on how tightly you coil it, and so on. Now, this shell, this is called a sundial shell from the Philippines, and it is basically a cone that's coiled. And if you coil a cone, you leave an empty space within the coils, and that space is called the umbilicus. You can see it right there on the underside of this sundial shell, that hole right there. That's the umbilicus. There are some snail species like this one. This is Solariella. It's an umbilical brooder. What Solariella does is lay its eggs inside the umbilicus, and it protects them there until they hatch. That's a spandrel. Snails didn't evolve coiled shells because the umbilicus was useful for egg brooding. They evolved coiling for some other reason, probably just to make their shape more compact. A snail that is basically um, I don't know, foot long, narrow spike is probably a lot more likely to break. If you can coil that up, you get a more compact shape that's probably more mechanically strong. I don't know exactly why snails evolved coiling. It may have been something like that. But the point is that the umbilicus is there because it has to be there. Because if you take a coil, there's a space in the center of the coil, right? If you take a slinky, that's a coil. In the middle of the slinky, there's a big open space. And the slinky's not there so that there can be an open space. The slinky's there because if you coil up anything, you leave a space in the middle. And it's the same thing if you're coiling a cone. If you coil a cone, you leave a space in the middle. And the idea is that Solariella uses that empty space for egg brooding, but that's not the reason Solariella has it. That space there exists because if you coil a cone, there's a space in the middle. It's there because of the geometry. It's there because it has to be there. Just like the spandrel has to be there if you put a dome on top of arches, the spandrel just appears, but it's good for displaying mosaics, but it's not the reason. That's not the reason why it's there. The reason it's there is because it has to be. So the umbilicus is a spandrel. It exists out of geometrical necessity. It's there because if you coil something, you leave a space in the middle. Using the umbilicus as an egg chamber is exaptation. Exaptation is taking a structure that's already present and using it for some new purpose that you weren't using before. And that's Gould's point. Natural selection does not make every piece of an organism adapt directly and perfectly to its environment. That's what Gould calls the adaptationist program. And it's very easy to tell what my old professor, Dr. David Wake, used to call just so stories, to come up with these terrific stories of how every piece of an organism was shaped by natural selection for a particular function, adapted perfectly to its environment. Gould argues that that's way too simple, that you have to look at other things, that, as he put it, a pluralistic view, one that looks at all kinds of causes, could put organisms back into evolutionary theory with all their recalcitrant yet intelligible complexity. Okay, so much for spandrels. Let me try and introduce one more idea in the time that we've got. I'm going to jump back in time to Ernst Haeckel. Haeckel was a German biologist, also a very good artist, and a pioneer in marine biology, especially of sponges, jellyfish, and deep sea critters. And he was also absolutely crazy about everything Darwin said, a great popularizer of Darwinian ideas in his native country. They used to call him Darwin's apostle to Germany. And his books have these very elaborate tree diagrams of how evolution has progressed. You've got Moneren down here at the bottom. That's like bacteria. And amoebas, Infusorientiere, that's like paramecium and things like that. The jellyfish are over here. The sponges are over here. The echinoderms are over here. There's the insects, far and away the biggest group of animals is this little side branch on the tree, segmented worms. But here we've got the sea squirts and the jawless fish and the bony fish and vogel, the birds and the reptiles. And here's the cetaceans and the carnivores and the bats and the apes. And here is humanity way up here at the top, right next to the gorilla, orangutan, chimp, and gibbon. Okay, so he draws these very realistic looking tree diagrams of how he thought evolution had progressed. He was also a pioneer in the study of embryology. His term for how an organism develops from egg to adult was ontogeny. And he's often quoted as saying ontogeny recapitulates phylogeny. What he actually said was the rapid and brief ontogeny is a condensed synopsis of the long and slow history of the stem, phylogeny. Phylogeny is evolutionary history. The cladograms and other things that you were drawing up are examples of phylogenetic trees. Phylon is ancient Greek for a tribe. Phylogeny is how tribes come to be, tribes and families and groupings of living things. And he argued that if you looked at the stages that an organism goes through as it develops from egg to adult, they reflect the evolutionary history of that organism. Not perfectly. There'll always be deletions and things like that. But looking at evolutionary history gives you the Cliff's Notes version of how that organism evolved from something much simpler. That the development of an embryo repeats its evolutionary history. He called that the biogenetic law. Here's one set of his diagrams. Up on the top, you've got early stage embryos of vertebrates: fish, salamander, tortoise, chicken, pig, cow, rabbit, and human. And you'll notice that at this very early stage, they don't have limbs. They've all got a, well, it's not quite, it says vertebral column. It's not quite the vertebrae yet at this stage, but they've got a central support axis surrounded by blocks of muscle. And they've all got this pharyngeal region, this region in the throat that's divided by little clefts, little slits, as it were. And the slits are separated from each other by arch-like structures called pharyngeal arches or gill arches. And there they are in the fish, these things right there. Salamanders have them, chicken, pig, cow, rabbit, human, you had these once. What happens later is that these embryos start becoming more different from each other. At stage 2, you can clearly tell the fish and the salamander apart. But all of these land vertebrates from tortoise to person look pretty darn similar, except for minor differences in proportion. It would actually be difficult to tell them apart. And at this stage, they've still got those pharyngeal arches. They're developing these paddle-shaped, flipper-like limbs. And then it's only later in development that they all become different enough from each other that you can start telling them apart. It's only later that the embryonic turtle develops a shell and looks clearly different from the chicken, and the embryonic pig develops a snout and looks clearly different from the embryonic human, and so on. The idea, Haeckel said, was that these stages that you're going through are repeating evolutionary history, that all vertebrates came from a critter that had a number of pharyngeal arches. And then all land vertebrates, all of this group, came from some critter that had pharyngeal arches and little flipper-like limbs. So these embryos, as you see them, are not perfect reflections, not exact copies, but kind of Cliff's Notes summary versions of what the ancestors looked like. That's the idea. Here's the thing. Haeckel was wrong in a very interesting way. Um, Haeckel wasn't really right about this, but he was wrong in a way that actually turned out to be pretty fertile. Um, he actually got brought up on charges before his university's discipline board for allegedly falsifying his drawings. I hesitate to say falsified, but he did simplify them a great deal, probably a little too much. For example, a chicken embryo at this stage doesn't look like this because it's actually lying flat on the egg, lying flat on where its belly would be, except it hasn't quite got one yet, and it doesn't really have this shape. Many of these embryos, you're completely leaving out the yolk sac. Human embryos don't quite look like this because of some quiddities in the way that they develop. You're leaving out the yolk sac and the amniotic membranes and all of that. A lot of these have been simplified a great deal. Certainly for these embryos down here, there's supposed to be a big, umbilical cord, which is not present in these drawings. They also vary a lot in size. So I honestly don't think Haeckel was really trying to cheat, but I do think his idea kind of got the better of him, and he oversimplified these diagrams a bit. The truth is more complicated. Haeckel's rival von Baer gave a more accurate summary that when you look at development, the features that are common to all members of a taxon appear before the features of the subgroups do. So the features that are common to like all vertebrates, like eyes, appear first, and then features that are common to all land vertebrates or tetrapods, like legs, appear next. And it's only after that that you get the beak of the chicken and the shell of the turtle and the snout of the pig and the hooves of the cow and so on. So von Baer's law is you go from features common to the larger group before you get features common to individual species. And that's probably more accurate, but even that has a lot of exceptions. Evolution can and does modify any stage of an organism's life cycle. There are cases, I know some sea urchins where the larvae look nothing like each other at all, and the adults, you can hardly tell them apart in different species. Organisms don't literally go through their ancestral stages. You never had an ancestor that looked like this. You did have ancestors that did have pharyngeal arches and clefts in between them. There are some very early ancestors of vertebrates, some are still around today, that sort of look like this, but not exactly, but maybe kind of if you squint and take lots of drugs, but not exactly. So you never had an ancestor that looked like that. You never had an ancestor that looked like this. Organisms don't literally repeat their ancestral stages. Having said all of that, Haeckel was right in that there's some very interesting connections between how organisms evolve and how they develop. And that study of the interrelationship has become a very fruitful area of research for about the past 30 years or so. People have been very keen on the field of evo-devo, evolutionary developmental biology. Vertebrates do, these are real vertebrate embryos, by the way. They go through a stage known as the pharyngula, at which they most closely resemble each other. Here you've got later embryos. There's a chicken with a beak. There's a cat with little paws. The bat is starting to develop wings, and there's a human embryo looking recognizably human. There up at the top, you've got earlier stage embryos, and they do look a lot more similar. They've got these tight flexes. It's hard to see, but they do have little pharyngeal slits right there with pharyngeal arches in between them. You did have pharyngeal arches in you at one stage. Um, and that's the pharyngula stage there. As I mentioned, Haeckel might have played a little fast and loose with his drawings, but at the pharyngula stage, the similarities are in fact real. What we now know is that the ways that different vertebrates get to the pharyngula can be radically different. Down here below, this is how the earliest vertebrate, or not even a vertebrate, one of the earliest chordates, the amphioxus, develops. It starts out as a hollow ball of cells, and then the gut forms when part of that ball pushes itself into the center. Chicken embryos develop in a radically different way, mostly because chicken embryos are so full of yolk that the chicken egg cells can't divide cleanly in two. And the upshot is that chicken embryos begin not as a ball of cells, like their distant cousins, the amphioxus, but as a disc of cells on top of the yolk. Chicken embryos, as they're laying down the very basic structures and forming their gut, look like discs. They don't look like balls. So you can diagram development across the vertebrates as being like an hourglass in time. In the earliest stages, here's time on the y-axis. Different stages can look very different from each other. As they develop, they go through this fairly narrow pharyngula stage where they look most similar to each other. And then after the pharyngula stage, they diverge to become, fish or chickens or people or whatever it is. So they start very diverse at fertilization, go through this narrow zone where they all look most similar to each other. That's the pharyngulas. And then diversify after that into the diverse adult forms that they'll eventually take. On Wednesday, I'm going to cover why this is so. Because over here, you might notice there's a little set of blocks right here that's been diagrammed, labeled HOX. HOX is a set of genes, and it is transcribed and translated at the phylotypic stage, at the pharyngula here. And embryos at the phylotypic stage are most similar to each other, not just in their shape, but in patterns of gene expression. They look similar because they're expressing genes in a very similar way. They're not necessarily expressing similar genes early, and they don't express similar genes late, but they do express the same gene set at this narrow point of the hourglass at the phylotypic stage. And we'll cover what those do in the next lecture on Wednesday. Okay, last thing I want to hit. Haeckel realized that one way you can get evolutionary change is to alter not so much the phenotype as the timing of development. That either the whole organism or one part of the organism can speed up or slow down developing relative to the organism's relatives. You can speed up or slow down the rate of development. You can start it earlier or start it later, or you can end it earlier or end it later. We don't need to go into all 6 of those, but they give you 2 basic classes. Paedomorphosis, pedo means a child, as in pedagogy, education, and pedophilia, which yeah, that means child. Paedomorphosis means shape of a child, where the descendant, either in whole or part, is less mature than its ancestor. The descendant looks like the juveniles of its ancestor. In paedomorphosis, the descendant is more developed than its ancestor, goes farther along in development than its ancestor did. And I'll show you a terrific example of paedomorphosis. We'll look at the axolotl. Okay, in the language of the Aztecs, that should be pronounced axolot, but I'm going to say axolotl because pronouncing it axolot, axolotl, trouble to go to. And it's native to central Mexico, where it's endangered, unfortunately. And it was first described as this really weird looking albino aquatic salamander that's got gills, external gills sticking out of its neck. Now, gills are a feature of larval salamanders, but axolotls are not salamander larvae because axolotls will reach sexual maturity and breed. You can keep a population of axolotls in a pond indefinitely. Well, somebody added iodine to the water where axolotls were living, and overnight he found that they transformed into adult tiger salamanders, Ambystoma tigrinum. They lost their gills, they bulked up their legs, and they started coming out on land. Axolotls are basically giant, overgrown tadpoles of salamanders that physically never mature, except their sex organs eventually do, and they reproduce and have babies while looking like overgrown tadpoles. So the axolotls are paedomorphic because they retain the shape and the structures of the juveniles of their ancestors, the tiger salamanders. Now, incidentally, some populations of axolotls will still turn into tiger salamanders when you add iodine. The water that they live in happens to be very poor in iodine. However, we now know that there are some populations of axolotl that have lost the ability to turn into tiger salamanders. They are permanently stuck as overgrown larvae. They can't become physical adults. So do we have one species or two? Well, that's actually kind of weird. But here's the critters that we're looking at. There's a tiger salamander up at top compared with an axolotl. You can see it's got these feathery gills and the swimming paddle on its tail. And there you've got a live one looking at you in the face. Axolotls are so grotesque looking that many people think they're adorable. They're so ugly, they're cute. So they're actually popular as pets, and you can raise, they're endangered in the wild, but you can breed them in captivity. So that's a case of paedomorphosis. The axolotl is a paedomorphic tiger salamander. And this gives us some idea as to how species like this evolved. Necturus, down below, that's the mud puppy. And we actually get these in, I've seen them collected in streams up in the Ozarks. They tend to like really cool, clear water. But we have these around here. They used to be popular for dissection in anatomy classes, although these days I think they're concerned about dropping populations. So I don't think Ben Cash has his students dissecting mud puppies in this day and age. But you'll notice that it's got gills, it's got relatively weak limbs, it's got a tail shaped like a paddle. Proteus up here, this is actually the European hellbender, is, if anything, even more so. You can see it's got gills, it's got underdeveloped limbs, it's got a tail shaped like a paddle. These are both paedomorphic salamanders. They retain features of salamander tadpoles, and they've completely lost the ability to metamorphose into anything else. They're permanently like this. These larval structures are retained throughout their lives, and hitting them with iodine now would do absolutely nothing to them. They're permanently paedomorphic. So that's an example of paedomorphosis for you. One more. These are the fetal skulls of two animal species that you might know. The fetal skulls look very similar to each other. And to visualize what's happening, I'm going to put a grid of lines over them. And there's that first species. As it develops from fetus to newborn to adult, the jaw and the face grow forward quite a lot, and you end up with this. This is a chimp, of course. That's what a chimp skull looks like. You can see the skull gets longer, but it doesn't get much higher. And the brows protrude and the jaws come out. The jaws grow rapidly compared to the brain case. And so you end up with a chimp. There's the second species, and I'll tell you right away that's a human. And that changes too. But notice how there's a lot less degree of change than the first species did. We still, our jaws get larger, our faces project a bit compared to our skull, to what we look like as fetuses. But the change is much less, right? A lot less change than here in the case of a fetal chimp developing into an adult chimp. So compared to chimps, we are paedomorphic. We look more like baby chimpanzees than we do adult chimps. This is probably true of our actual ancestors. We're not descended directly from chimps, but we're descended from apes about 5 million years ago that would have looked kind of chimpy. And one of the main reasons why we're different from them is we are comparatively paedomorphic. Our brain expands for a much longer period. Our facial expansion is slowed down. And the ultimate result is that we look like we are chimps that didn't finish growing up. We are closer to baby chimps in appearance than we are to adult chimps. The very last thing I'll mention is that paedomorphosis, right? Descendant looks like a juvenile of its ancestor. In mammals, that's often considered to be cute by humans. Baby mammals tend to have a high forehead, large eyes relative to body size, a small jaw relative to head size, and a big head relative to body size. These are all features that you see in human babies, right? Big forehead, big eyes, little jaw, big head on a small body. And it has been suggested that humans are behaviorally predisposed to react to anything that looks paedomorphic with feelings of caring, nurturing, and protection. For the simple reason that somebody who thought babies and children looked repulsive is not going to reproduce very much, right? If you think babies are hideous and you just want to drop kick them, you're not likely to have many surviving offspring. If you think babies are adorable and you want to protect them, you are likelier to have offspring. And the evolution of human behavior, so the idea goes, extends not just to human babies, but anything that triggers your awe feeling in your brain with a big forehead and little eyes and a big head and a little jaw. So there you have humans at different ages who have all been drawn to the same scale. As an adult, your head is about 1/8 of your total height. The height of your head is about 1/8 of your total height. A newborn, the head is about a quarter of height. So you can see that your relative to newborns, your head takes up a smaller proportion of your total body size. Newborn limbs are shorter, bodies are smaller, foreheads are much bigger. Your forehead is roughly half of your head size as an adult, but it's easily more than half as a newborn, right? That right there, yeah, the distance from the top of your head to your eyes is more than half the length of your head as an adult. Is more, sorry. Okay, as a newborn, the distance from your top of your head to the line connecting your eyes is more than half the total length of your head. Whereas as an adult, it's almost exactly half. So babies have bigger foreheads and bigger eyes and bigger heads on smaller bodies and shorter legs than adults do. And the idea is that that is what we cue on when we think that something looks really cute. So, for example, there's an adult dog. That's a pretty dog, but the puppy has a higher forehead and bigger eyes relative to its head and a smaller jaw. And you were probably looking at that puppy and going, oh, he's cute. Kittens, it's the same thing. They've got big eyes, high foreheads, large heads, and short legs. And this turns up a lot in the history of animation. On the left is The Simpsons in 1987. They began as short films shown on a variety show of the time called The Tracy Ullman Show. The actual show began in the Simpsons TV show began in 1989, as I recall. It was weird. I spent 89, 90 in Scotland, and I came back, and everybody is wearing these Bart Simpson t-shirts with this guy saying, "Don't have a cow, man," and I can't figure out what's going on. Notice the difference. Bart on The Simpsons has a high forehead and a small jaw and large eyes relative to body size, and he looks like an adorable scamp. 1987 Bart has a low forehead and a big jaw and relatively small eyes relative to body size, and he looks like he tortures squirrels for fun. Homer Simpson in 1987 has this sloping low forehead and big jaw and small eyes, and he just looks mean. Homer Simpson in The Simpsons Show has a straighter forehead and relatively smaller jaw and definitely bigger eyes, and he looks like the adorable alcoholic goof that we've all come to know who says, "Do." You know, Lisa over here looks smart. Lisa over here looks like she's gonna get knocked up at 13. Um, you know, Marge looks a lot cuter in the show than she did back in 1987 because she's got a smaller jaw and bigger eyes than she used to. So the characters on the Simpsons show now are a lot more paedomorphic. And notice how this changes your emotional reaction to them. You tend to react more warmly to more paedomorphic looking characters, even if they're not drawn all that realistically. You know, even if Bart's head looks like a coffee can, you still react to him, you know, more warmly with a straight forehead, a high, large forehead, and a small jaw, as opposed to your reaction to him with a big jaw and a small forehead. You know, this Bart looks like he's going to burn down your house because he thinks it's funny. That Bart in the show looks like he's going to get into um, charming, childish scrapes and say "ay caramba" a lot, which is what he does, as I recall. So we'll stop right there, leaving paedomorphosis aside, and we'll get into some molecular nitty gritty on Wednesday. And I will now stop the recording.